math to give you guys an introduction to biology and the greater context of astrobiology. So we can think about the biologist's contribution or their utility to astrobiology, and I think this is a really interesting question because I really don't think that the full potential of the biologist has been realized yet, and that's really for sort of the plain reason that we haven't really found anything yet. So we haven't found any of these guys or something like this. We haven't really found any life forms, extraterrestrial life forms that the biologist could then go in and categorize and characterize. But they can still do some things. So one of the things biologists can do in the context of astrobiology is look at the origin of life on Earth and what that informs us about the origin of life elsewhere. So obviously the origin of all life way back, single cellular life, but also evolution throughout time. So eukaryotic life, multicellularity, large macrofauna, and then even the evolution of things like intelligence in biology. And so a few of the talks we're going to hear today are going to cover that topic. Most of them are going to center around here at the beginning. But Valerio is going to tell us about the evolution of biopolymers. And so molecules important to biology mostly exist as polymers, so things like DNA and protein. So these strung together chains of monomers. But what's interesting is that a lot of the monomers that make up present-day biopolymers are sort of really hard to assemble under prebiotic conditions. And so a lot of people think that the monomers that we know and love today are actually descended from sort of these proto-monomers that made up protopolymers of similar but fundamentally different molecules. So Lara is going to tell us about that. Jose is then going to tell us about the origin of viruses. And so this is a really interesting topic because I don't think it's one a lot of people think of. Everyone's so obsessed with finding the origin of cellular life, whereas we have these viruses that have a huge bearing on curbing biological populations down from single-cell bacteria all the way up to humans. These things that are technically non-living acellular life forms who have genomes that not only DNA like all cellular life, but also RNA. And so trying to pinpoint how these things evolved, why they evolved, is a very interesting question in terms of life as we know it, or as we may not. So then finally, under the umbrella of this topic, Anna is going to talk to us about membrane formation. And this is a really important step in terms of going from prebiotic chemistry to cellular life, because sort of we can think of without a membrane, we sort of have this diffuse prebiotic chemistry where things are far apart, they're not high concentration. Molecules that are reacting with each other might not be doing so in a very fast fashion. But then, if you can enclose this in a membrane, you're concentrating and compartmentalizing these molecules so that they can interact a lot more commonly and a lot faster with each other. So you're sort of exponentially then increasing from that point, getting to sort of something we would recognize as a modern life form. So origin of life. Another thing biologists can do is extraterrestrial analog studies. And so what I have here is just the Earth, and obviously we can think of this as environment. There are environments on Earth that are analogs for other planets, either in our solar system or even exoplanets, things like that. Because of the geochemical, the atmospheric conditions in these environments, they mimic, possibly something like Europa or Mars. This can also apply to organisms themselves. So you can have an organism that because of the metabolism it employs, it is an analog. It might not necessarily live in an analog environment, but it could be an analog for an organism we might see on another environment. For instance, perchlorate reducers might not all live in Mars analog environments, but that might be a very interesting metabolism for something that would be living on Mars right now. So Lara's going to talk to us about analog environments, and she's going to talk about the Atacama Desert in the Andes. You can just see basically, the picture. This is Earth and this is Mars, obviously, but very similar. And so what we do in these analog environments is basically we go in and we look at who's there and what they're doing. So we first look at who's there, and then probably more importantly, how are they making their living there? How are they utilizing what's there to grow, to make necessary molecules and divide? So those two, and then another one that maybe isn't as thought of, but that Larry DeLucas really introduced very well, is that the biological effects of spaceflight in the space environment. So something like in a microgravity environment, how that affects organisms all the way up from humans down to bacteria. And so that's a really interesting question, because this is an environment that nature and evolution have never seen and have never had to plan for. So understanding, how it adapts to this environment or what changes happen is a very interesting question. So Michael's going to talk to us about that with this model gram-positive bacterial organism called Bacillus subtilis. And he's basically just going to look at gene expression, comparing that to, let's say, we have a cell where gene Y is turned on and gene Z is turned off on Earth, but then we put it up in the International Space System, and then under all conditions being the same, except for the fact that it's now in space, well, gene Y is turned off and gene Z is now turned on. And so why does that happen? So then finally, and it sort of goes along with this third bullet point, is we have planetary protection, human colonization of planetary bodies. So there's a reason NASA spends all this money to make these things that they're sending out into space extremely sterile, extremely abiotic. That's because we're very worried about bringing things like microorganisms from Earth and landing on an environment Mars where they might be able to grow. And now we have basically compromised the planet by bringing earthbound bacteria to that. And so there's a lot of research looking at how basically we can keep from messing up the planet, but then also how we can, while trying to not do that, form settlements or stable colonies on these planets, both sort of self-contained ones on places like the Moon or Mars, and then also possibly on planets that might have atmospheres that are conducive to human life, open settlements. And I don't think Drew going to make it today for this talk, but she was going to give this really interesting talk about basically looking at how humans on Earth through time have affected their environment, both closed systems, natural systems that aren't as affected by humans, and then other open systems. And really what that means for our ability to basically sustain something like some type of dome on colony on Earth or on the Moon or Mars, but also some type of actual new settlement, uncontained settlement on an exoplanet that might have an atmosphere or an ecosphere similar to Earth. So with that, I'd just like to invite you to sit back, relax, and enjoy what I'm sure will be some fascinating and very thought-provoking talks. That's all I got.